So these are ideal characteristics that we want to have a good, efficient antimicrobial agent. So the mechanisms by which antimicrobials work are kind of wrapped around these 5 targets or 5 strategies that are used. The first one is to prevent the formation of the cell wall in bacterial organisms. So we have a group of antimicrobials or a group of antibiotics known as cell wall synthesis inhibitors because they inhibit the production of the cell wall in the bacterial organism. The next mechanism we have is the strategy to disrupt or to damage the cell membrane, which lies just below the cell wall of a bacterial cell. The next group is what we would refer to as nucleic acid synthesis inhibitors, which means we produce an antibiotic treatment that is used to destroy DNA or RNA synthesis, or plays a role in disrupting that process, or any of the processes that take place in the central dogma theory. And we'll talk a little bit more about those in just a second when I blow up this figure. But the next group are protein synthesis inhibitors. And these really just go after different proteins that are inside the bacterial cell, and the drug treatment starts to denature those proteins, or it starts to block the process of translation, which gives rise to a protein. And once you block that process, you inhibit it, which is why we refer to these groups of antimicrobials as protein synthesis inhibitors. And then lastly, we have another common mechanism, which is known as a group of treatments that are known as anti-metabolites. They are anti the metabolism of the bacterial cells. So what they do is they target, these particular drugs will target enzymes and disrupt enzyme activity as is taking place in the cell. And this diagram is a nice diagram that kind of outlines, I'm going to move this, outlines those different types of strategies. There we go. All right, so in here you can see examples of the first group or common mechanism, which is that of the cell wall synthesis inhibitor. And we have some example drugs that are included in this group, such as penicillins, cephalosporins, vancomycin, bacitracin, isoniazid, as well as ethambutol. And what happens here is these particular treatments or these particular drugs have a mechanism in place by which they're able to disrupt the cell wall. Penicillin has a beta-lactam ring structure, which is used as kind of a drill to drill its to take the ring and drill the ring through the gram-positive bacterial cell wall. And when it does that, all of the solutes on the outside of the cell start coming into the cell, and that causes the cell to swell, and then the cell will start to burst or lyse. So that's one mechanism that's used by cell wall synthesis inhibitors, is basically taking that cell wall and perforating it, causing a change in osmotic pressure, which causes the collapse of the cell. The next group that we talked about was cell membrane disruptors. A cell membrane, it doesn't look like on this diagram they give you too much to go by, but you can see here at the base of the cell wall. The cell wall is this red area here, which has the peptidoglycan embedded in it, and then just around the cytoplasm of the cell and below the base of the cell wall, you have the cell membrane. And so, as if you have a treatment that's targeting structures in the cell membrane, this is the structure that's going to get disrupted. You can interrupt a pathogen's ability to attach itself to a particular location or to a surface or to the host, there's a treatment that's called Aerodon, which is used to help disrupt that attachment. And disrupting the attachment obviously is going to keep the bacterial cell from adhering to the tissue to induce infection. Nucleic acid treatments, which we discussed in here as a third mechanism for antimicrobial agents to use. This is inhibiting DNA and RNA production. So you're attacking, again, those processes of the central dogma theory, such as DNA replication or messenger RNA transcription. And some examples that are used here as treatments are actinomycin nucleotides. You can also use some analog treatments, quinolones and rifampin. They're very specialized and they target different processes of DNA or RNA synthesis to prevent synthesis from taking place. Oh, here is, I'm sorry, I didn't see this. This particular diagram here is the cell membrane. Remember we told you the structure here was this yellow area. So this is another, this is essentially the damage to cell membrane example. And one of the key treatments that's used here are called polymyxins. And so polymyxins will start to disrupt the phospholipid bilayer of the cell membrane. And in disrupting that, that causes the cell to break down. Protein synthesis, as we talked about. Some example treatments are tetracyclines, chloramphenicol, macrolides, and aminoglycosides. And these particular treatments are targeting specifically the ribosomes that are instrumental in producing proteins. The ribosomes of bacterial organisms are different than the ribosomes found in human cells. And because of that difference, we're able to target specifically bacterial ribosomes with treatment plans. The last group, remember, were the antimetabolites. These inhibit enzyme activity, and then they can prevent certain metabolic pathways from taking place inside of the cell. Some examples are trimethoprim and sulfamides. Oops. Okay, so like I said, there's a lot of different common mechanisms that can be used. And one of the reasons we have these different mechanisms is because bacteria can, obviously develop drug resistance. And when they develop drug resistance, we have to figure out some alternative ways to destroy these guys, as well as producing antibiotic treatments that are going to be more widely used by everybody versus creating just one common attack mechanism and then producing a treatment in which some people might be allergic or some people don't respond well to it. Okay, and so as I said, there's some different types of drugs that are out there that target different things. Remember we talked about the key terms bacteriostatic and bactericidal. Some drugs simply inhibit the growth of bacteria, which means they don't necessarily kill the bacteria, but they release some sort of control mechanism that prevents bacteria from growing. And so the bacteria aren't able to grow anymore, so eventually they will die off, but more importantly, they're unable to replicate. So bacteriostatic drugs are drugs that make bacteria static. And by static, it means they can't grow anymore. They're just, they're just stuck where they are. Bactericidal drugs, though, they will produce elements that are used to actually kill the bacterial cells. So by destroying the bacterial cells, you eliminate the pathogen. And again, we kind of talked about the pros and cons of this earlier, where if you kill bacterial cells, sometimes you could produce, some toxins inside of the host tissue that could be more problematic to the patient. And so one drug I do want to talk about is that of sulfamide, which is a drug that's used to prevent the formation of a vitamin known as folic acid. And the folic acid is used by bacteria in order to help them to grow and develop. And they can't produce certain proteins. So in a sense, it's a protein inhibitor. But with the sulfamide drugs, there's a specific type of acid that the bacteria need to produce called p-amino benzoic acid. And so if this acid is not produced, then the bacteria can't function. So what happens in this particular process is that the sulfamide is acting as what we call a competitive inhibitor. And it basically means that it is serving as a blocking mechanism to prevent the bacterial enzymes from joining where it needs to. Okay. So the normal process that happens in a bacterial cell to produce folic acid is the use of this, what we call this PABA protein. Okay, this PABA protein is going to lock in to this specific enzyme here. And when that happens, that allows folic acid to be produced. So this here is our normal mechanism that takes place. But what we can do when we produce the use of a sulfamide to inhibit this process, we block the binding of this PABA structure to this particular enzyme. Okay, so instead, so you see the PABA protein is this C double bound to O bound to OH, but instead of allowing PABA to bind here, you have a different particular substrate that binds to this particular enzyme. And so you see that we have a new molecular or chemical equation, which is oxygen double bound to sulfide double bound to another oxygen bound to an amino group. And so this blocks this PABA protein from binding to this enzyme, and in doing so it blocks the formation of folic acid. So this is why we talk about sulfa drugs as being competitive inhibitors, because they're not really doing anything other than taking up space, and they're blocking the actual PABA protein from locking into the enzyme in order for it to function, to generate folic acid. So that's how that particular process works. And this is going to cause not the death of bacteria, but it will inhibit their growth because now they can't produce folic acid. So that's how that works. So again, bacteriostatic drugs are going to inhibit the growth of bacteria, and then bactericidal drugs will kill bacteria. Penicillin, as I said before, typically will target the cell wall because it uses what's called a beta-lactam ring structure. And the penicillin will start to destroy the cross-linking of the peptidoglycan that surrounds the cell wall of the bacterial cell that's embedded in the cell wall. And once that peptidoglycan has been disrupted, then the gram-positive bacteria is going to be degraded, or it's going to lyse or rupture, like I was saying earlier. And so you can see in this image, here you have a bacterial cell, and then you have penicillin. Penicillin makes its way to the bacterium, and it locks in the bacterium. And once it attaches to the bacterium, it will drill a hole using that beta-lactam ring structure and cause the cell to lyse. And that's essentially what this figure is showing you here. Just a little bit bigger. I like these figures that are transparent. But what this is showing you here is this is the peptidoglycan structure. Okay, so peptidoglycan is made of these NAG&NAM units and then held together by different amino acids. So that's what these little balls looking things are. And so you have these structures that are known as a penicillin binding protein, and they start locking